We spend a massive amount of time on this show, and I mean, in biology in general, just completely obsessing over the brain.
What's the control center, right?
It is, yeah. We talk about signals and nerves and neurotransmitters, all the whispers between cells. We really treat the body like it's this giant communication network.
Which, to be fair, it is.
It is. But I had this flash of insight while I was going through the source material for today. You could have the most brilliant brain in the universe, sending out the most complex, poetic signals you can imagine.
Okay.
But if you don't have a way to translate the electricity into physical movement, you're just a ghost in a shell. You're a brain in a jar.
That is a pretty terrifying image, but you are absolutely right. The nervous system is totally useless without an effector. It needs a way to actually, impact the physical world.
And that is what we are doing today. We're crossing that threshold. We're going from thinking to doing. We are talking about the machinery of action. We are talking about muscle.
Finally. And I'm glad you framed it that way because muscle isn't just meat. It's a transducer. It is a biological machine specifically designed to take a spark of electricity, an intention, and turn it into mechanical force.
A squeeze.
A squeeze, exactly.
And to guide us through this incredible machine, we're looking at two absolute titans of the field. First, we've got Ganong's review of medical physiology.
Ah, Ganong.
And I'm realizing Ganong is sort of the engineer of the group. It's very focused on the molecular nuts and bolts.
Oh, absolutely. Ganong is incredible for that cellular depth. If you want to know which specific ion channel opens at exactly which millivolt, that's your book. It gives you the high-resolution blueprint.
And then we have the legendary Guyton and Hall textbook of medical physiology.
The mechanic.
Mechanic, I like that.
Guyton is all about flow and systems and the why. Why does the system behave this way? How does it all integrate with the heart, the blood, the lungs? It's the big picture.
So today, we're really synthesizing the excitable tissue muscle chapters from Ganong with the skeletal and smooth muscle chapters from Guyton. Our mission, really, is to get the logic of it. How does the body convert that spark into a squeeze?
And more than that, how does it build three completely distinct engines, skeletal, cardiac, and smooth muscle, using the same basic machinery?
Exactly. Before we dive into the molecules, I want to set the stakes with that very question. Because we aren't just memorizing names of proteins today, we're trying to solve a puzzle.
A physiological puzzle.
The body has to do three completely different jobs with basically the same toolbox. It needs to sprint, that's explosive voluntary power.
Skeletal muscle. Which needs to be incredibly fast, but we accept that it gets tired.
Right, I can't sprint forever. That's the trade-off. Then you have the heart, cardiac muscle. It needs to pump reliably, rhythmically, 24/7 for maybe 80 years or more.
It is not allowed to take a break.
Not for a second. And it is absolutely not allowed to cramp.
If the heart cramps, you die. It's really that simple.
And then you have that third engine, the quiet one, smooth muscle, your stomach, your bladder, your arteries. These guys need to hold tension, sometimes for hours or days, without burning through all your energy.
It's the efficiency engine. The marathon runner of the group.
So the central question for our deep dive today is how? How does the body take the same core ingredients, calcium, ATP, actin, myosin, and build a drag racer, a reliable pump, and a fuel-efficient cargo ship?
That is the perfect framing. It all comes down to structural trade-offs, speed versus endurance, voluntary control versus automaticity. Form follows function.
Okay, let's pop the hood. We need to look at the engine block itself. If I take a piece of skeletal muscle, let's say from my bicep, and I zoom in way past the fiber bundles, past the cell membrane, what's the fundamental unit I'm looking at?
You are looking at the sarcomere. If you look at skeletal muscle under a microscope, the first thing you notice are these stripes, striations, dark bands, light bands, dark bands, light bands.
And that's not just a pattern, right? It's real.
It's the architecture. It is the physical structure of the sarcomere repeating over and over again.
Ganong describes this with such intense geometric precision. It honestly feels less like biology and more like crystal lattice work or something.
It's highly crystalline. It's incredibly organized. You have these Z lines, which act as the borders. And inside, you have this repeating structure of filaments. And this brings us to probably the most famous visual metaphor in all of physiology.
The interlaced fingers.
The interlaced fingers, right. If you're listening, hold your hands out in front of you. Spread your fingers. Now, slide the fingers of your right hand in between the fingers of your left hand.
Okay.
But don't let your palms touch. Just let the fingers overlap a bit.
Got it. I'm doing it.
Okay. Your left hand represents the actin filaments. These are the thin filaments. You can think of them as the rope. They are essentially passive in this interaction. They're anchored to the Z lines, which would be like your left wrist in this analogy.
So actin is just there to be grabbed and pulled on.
For the most part, yes. Now, your right hand, that represents the myosin, the thick filaments. This is the motor. This is the molecular engine that does the work.
Okay, so when I flex my bicep, what's happening in this analogy?
Your right fingers, the myosin, are crawling up your left fingers. The actin, they're pulling your hands closer together. And here's the critical point. The filaments themselves don't shrink. Your fingers don't get shorter.
The overlap increases.
Exactly. The overlap increases. This is the sliding filament theory.
And Ganong points out a detail that proves this, which I found really clever. When you look at those bands under a microscope, the A band, that's the dark stripe where the myosin is, it never changes with.
It stays constant.
It stays constant because the motor isn't shrinking. It's the Z lines, the borders that are moving in. The whole room is shrinking, but the furniture inside stays the same size.
That's a great way to put it.
I want to pause on that furniture for a second. The myosin, in our hand analogy, it's just fingers. But Ganong gets really specific about the shape of this molecule. It's not just a simple stick.
No, and this is absolutely crucial for understanding how the force is generated. Myosin is this complex protein made of heavy chains and light chains. The heavy chains wrap around each other to form a long tail. That's the structural backbone of the thick filament.
But the action isn't in the tail.
No, the action is in the heads. Sticking out of this tail, you have these globular heads.
Heads, right.
Think of them like the head of a golf club, but it's attached to the shaft by a flexible hinge. And there isn't just one. There are hundreds of these heads along the filament, spiraling around it. It's like a corn cob where every kernel is a tiny hinged hand reaching out.
And Guyton mentions another really important protein that keeps this whole thing from falling apart, titin.
I love titin. It's one of the largest protein molecules in the entire body. It's gigantic.
It sounds like a titan.
It basically is. It's this massive springy scaffold. Its job is to anchor the myosin to the Z line, so it keeps the motor centered in the sarcomere. Without titin, the myosin would just float away or get crooked when the muscle contracts and relaxes.
So titin is like the suspension system.
It's the suspension system, and it also provides passive elasticity. When you stretch your hamstring and you feel that resistance, a lot of that is titin fighting back, trying to pull the sarcomere back to its resting length.
Okay, so we've got our parts list. We have the rope, which is actin. We have the motor, myosin, and the springy suspension, titin. Now we have to make it move. We have to talk about the cross-bridge cycle.
The rowing mechanism.
Guyton calls it the walk-along theory. But frankly, when I read the chemistry, walking feels way too peaceful. This is a violent, high-energy event.
It is violent. It's a molecular ratchet, a series of tiny explosions.
So walk us through the cycle, step by step. Because there is a paradox here involving ATP that I think confuses almost everyone the first time they hear about it.
It is the single most counterintuitive part of muscle physiology. But once it clicks, it makes perfect sense. So let's start at step one, attachment.
Okay.
Myosin head, which is in a high-energy cocked position, extends and binds to a specific active site on the actin filament.
It grabs the rope.
It grabs the rope. Now, step two, the power stroke. This is where the shape change happens. The head tilts. It bends at that hinge we talked about. This physical bending drags the actin filament along with it, maybe about 10 nanometers.
Like rowing an oar. You plant the paddle in the water and you pull.
Exactly. And this stroke releases the energy that was stored in the head. It kicks off a phosphate and an ADP molecule that were attached to it. Snap. Motion occurs. The sarcomere shortens by a tiny fraction.
Okay, so now the head is bent, it has pulled the rope, and it's still holding on tight.
Right. It is latched on. This is called the rigor complex. It's a very stable, low energy state.
Now, intuition says, okay, to pull again, I need to use energy to squeeze. But that's not what happens, is it?
No. This is the paradox. To let go of the rope, to detach the myosin head from the actin, a fresh new molecule of ATP has to swoop in and bind to the myosin head.
Wait, so I have to pay energy to relax? Or to let go, at least?
Yes. The binding of ATP causes another conformational change in the myosin head that makes it lose its grip. It releases the actin. If you don't have ATP, you cannot let go.
That is wild. We usually think of effort as the contraction and relaxation as just stopping the effort. But molecularly, the off switch is an active, energy-consuming step.
It is. Once that new ATP binds and the head detaches, the story isn't over. That ATP is then hydrolyzed. It's broken down into ADP and phosphate.
And that releases energy.
That explosion of chemical energy is used to recock the head. It pushes it back to its upright high-energy position, ready to grab the next run of the ladder and start the cycle all over again.
So the resting state of a muscle when I'm just sitting here is actually a state of high potential energy.
Exactly. Your myosin heads are all cocked, loaded, and waiting. They have the energy stored in them, like millions of set mousetraps.
And the only reason they aren't snapping right now is because, well, we'll get to the safety catch in just a minute, but I want to really nail this ATP to detach concept because there is a gruesome but perfect real-world example of what happens when you run out of ATP.
Rigor mortis.
The stiffness of death.
This is the ultimate proof of the mechanism. When a person dies, breathing stops. Oxygen delivery stops. The cells can't perform aerobic respiration, so they can't make any more ATP.
So what happens inside the muscle cells?
Well, a few things. First, the calcium pumps, which we'll talk about, also need ATP, so they fail. Calcium leaks out of its storage tanks into the cell. The myosin heads see the actin binding sites are now exposed, and they grab on.
They perform one last power stroke.
One last power stroke. But then there is no fresh ATP molecule to come in and tell them to let go.
So they're stuck, frozen.
They are chemically welded to the actin. Every muscle in the body becomes rigid. It locks in place. And it stays that way until the proteins themselves start to physically decompose a day or two later.
That is incredibly macabre, but it perfectly illustrates the point. Relaxation, or at least detachment, is an active metabolic process. You have to buy your relaxation with ATP.
And this leads to an interesting efficiency principle that Guyton discusses, called the Fenn effect.
The Fenn effect. Is this just a fancy way of saying more work equals more gas?
Basically, yes, but it's chemically responsive, which is the cool part. The muscle is smart. It doesn't just burn ATP at a constant rate, regardless of what it's doing.
Okay.
The more work the muscle performs, the heavier the load it's lifting, the more ATP is cleaved. The cross-bridge cycle actually speeds up. If the myosin head tries to pull, but the load is too heavy to move, the cycle slows down. It doesn't just spin its wheels, burning fuel for no reason.
It's like a smart throttle that automatically adjusts fuel consumption based on the demand. Okay, so we have the engine. We understand the pistons, the sliding filaments. We understand the fuel ATP and its weird paradoxical role. But an engine doesn't just start itself. If my myosin heads are all cocked and ready to go, why aren't my muscles in a constant state of spasm? What is the safety catch?
The safety catch is the troponin-tropomyosin complex. And honestly, without this beautiful little piece of molecular engineering, we would be, just gelatinous blobs of constant spasm.
So describe this blockade. What does it look like?
Okay, picture the actin filament again, the rope. It's actually two strands twisted together, like a helix. Wrapped around this rope, sitting in the groove of the helix, is another long, thin, wire-like protein. That is tropomyosin.
And what is it doing there?
It is physically sitting on top of the myosin binding sites. The myosin head is right there, wanting to grab the actin, but the tropomyosin is in the way. It's like putting a strip of tape over the Velcro.
So it's a physical barrier. How do we peel the tape off?
We need the key, and the key is calcium. And the lock that the key fits into is another protein called troponin.
Gannone breaks troponin down into 3 subunits, and I think this actually helps clarify its function. Troponin I, T, and C.
Right. It's a complex of 3 parts. Troponin T is what tethers the whole complex to tropomyos, the wire. Troponin I is the inhibitory part. It helps hold the wire in the blocking position.
But troponin CC for calcium.
That's the sensor. Troponin C has a high affinity for calcium ions. It's got binding sites waiting for them.
So when calcium finally shows up on the scene,
it binds to troponin C. This binding causes a conformational change of shapeshift. The whole troponin complex twists, and because it's attached to that tropomyosin wire, it physically drags the wire out of the groove.
It rolls the blockade out of the way.
It moves the tape. The binding sites on the actin are now exposed. The cocked myosin heads that have been waiting this whole time immediately snap onto the actin, and the power stroke begins.
So calcium is the go signal.
Calcium is the only signal that matters inside the cell. It is the final common pathway for contraction. If intracellular calcium is high, you contract. If intracellular calcium is low, you relax. End of story.
Which brings us to the biggest logistical problem in muscle physiology. Where does the calcium come from, and how do we get it there fast enough to be useful? This is section 2, excitation-contraction coupling.
This is this spark in the switch, the link between the nerve and the filament.
Here's the problem as I understand it. A single muscle fiber is huge. On a cellular scale, it's not a microscopic little blob. It can be centimeters long and very thick. If a nerve signal, an electrical wave, hits the outside skin of the muscle,
The sarcolemma.
Right. If it just hits the sarcolemma, how does the very center of the muscle, deep inside, know to contract at the exact same time? Diffusion is slow. If I drop a bit of ink in a bathtub, it takes minutes to spread out. We need the entire muscle to fire in milliseconds.
Diffusion is way, way too slow. If we relied on diffusion from the surface, the outside of your muscle fiber would contract long before the inside even got the message. You'd tear the fiber apart from the inside out. It would be a disaster.
So what's the body's solution? How do you get a signal from the surface to the core instantly?
Plumbing. Hardwired high-speed plumbing. The T-tubules.
T for transverse.
Exactly. The cell membrane, the sarcolemma, actually invaginates. It folds in on itself, forming these long, thin tunnels that dive deep into the core of the muscle fiber, right down to where the filaments are.
And Guyton makes a point to emphasize that these tubes are actually open to the outside world. They contain extracellular fluid.
That's a key point. It's not a closed internal system. Think of it like sticking your finger deep into a big ball of dough. The surface of the dough, your skin, is now technically deep inside the ball.
I see. So when the action potential, the wave of electricity sweeps across the surface of the muscle cell, it doesn't just stay on the skin.
It dives down the T-tubules. It propagates right to the center of the fiber at the same speed, ensuring the entire cross-section of the fiber gets the go signal at the exact same millisecond.
Okay, so the electricity is now deep inside, but the calcium, the actual key, is stored in a separate tank, right?
Right. It's stored in the sarcoplasmic reticulum, or the SR. This is an intricate, web-like network of hollow tubes that's wrapped around all the myofibrils. You can think of it as a calcium vault.
And the concentration inside is huge.
It's immense. The concentration of calcium inside the SR is about 10,000 times higher than it is in the cell fluid, the cytosol. It's a loaded weapon.
So we have the signal, the electricity, in the T-tubule running right alongside the vault, the SR, which is full of calcium. How do we connect them? And this is where Ganong details a mechanism that is basically just mechanical engineering. It's brilliant.
This is one of the coolest and most unique parts of skeletal muscle. It involves two receptor proteins with typically terrible names.
The dihydropyridine receptor, or DHPR, and the ryanodine receptor, RYR.
Physiologists are just terrible at naming things. Let's simplify. The DHPR is on the T-tubule membrane. It is a voltage sensor. Its job is to detect the electricity coming down the pipe.
And the RYR?
The RYR is on the sarcoplasmic reticulum membrane. That is the floodgate. It's the channel that holds the calcium back.
And in skeletal muscle, what's the relationship between them? They touch.
They are mechanically coupled. They are physically linked together. This is so important. Think of the RYR on the SR as a plug in a bathtub drain. The DHPR on the T-tubule is physically attached to that plug.
Okay.
When the electricity of the action potential hits the DHPR sensor, the sensor changes shape. And because it's attached to the plug, it physically yanks the RYR, out of the drain.
That is so distinct. It's not sending a chemical messenger across a gap or anything like that. It's a physical pull.
It's a direct mechanical pull. The plug opens, and calcium explodes out of the SR down its massive concentration gradient. It floods the sarcomere, binds to troponin C, moves the tropomyosin tape, and boom, contraction.
And then to relax, you have to put the genie back in the bottle.
And that is hard work. That's an active process. You have a pump called the Sarca pump. It stands for Sarcoplasmic Endoplasmic Reticulum Calcium ATPase.
Another great name.
It's a vacuum cleaner. Its only job is to suck calcium from the cytosol back into the SR against that 10,000-fold concentration gradient.
Which uses ATP.
Lots and lots of ATP. This is a huge reason why you generate so much heat when you exercise. A massive portion of your metabolic energy is just running the Sarca pumps to clean up the calcium and reset the system for the next contraction.
Before we move on from this mechanism, we have to talk about what happens when this plumbing fails. Guyton mentions a terrifying clinical scenario, malignant hyperthermia.
Oh, this is a true anesthetic nightmare. It's a genetic condition, usually silent until it's triggered.
Right. So imagine you're an anesthesiologist. You have a patient on the table for a routine surgery. You give them a standard inhaled gas anesthetic, like halothane. Suddenly their jaw locks shut. It becomes rock solid.
Masseter spasm. It's a classic early sign.
Then you look at the monitor. Their end-tidal CO2 is skyrocketing. Their temperature is climbing, not slowly, but spiking. They are literally cooking from the inside out. What is happening on a molecular level?
That anesthetic gas has triggered a defect in their ryanodine receptor, the floodgate. The plug got pulled and now it's stuck open. It can't close.
So the calcium is just pouring out continuously.
Uncontrollably. So the muscles are contracting maximally all over the body. That's the rigidity. And the circa pumps are running at full blast, screaming, trying to suck the calcium back in. They're burning through ATP like crazy.
And that generates the heat.
Massive heat, that's a hyperthermia. And all that ATP burning creates enormous amounts of CO2 and lactic acid, that's the acidosis.
And the muscle cells eventually just break down.
They literally run out of energy and die, releasing their contents into the blood. It's called rhabdomyolysis. And the patient will die if you don't intervene instantly with a specific drug called dantrolene, which blocks that leaky channel.
Wow. It just shows you that these textbook diagrams of ion channels and pumps aren't just abstract theory. If that RYR valve is slightly sticky in the presence of a certain chemical, it's a fatal flaw in the machine.
It's a very sobering reminder of how exquisitely precise this regulation has to be.
Absolutely. Okay, so that's the mechanism. We have the motor and we have the ignition. Now let's talk about performance. This is section 3. Skeletal muscle mechanics. Because a muscle doesn't just contract yes or no, it has nuance. It has to be graded.
Right. It has to be able to lift a feather, and it has to be able to squat 300 pounds. The same muscle has to do both.
One concept that I found fascinating in both Ganong and Guyton is the length-tension relationship. It suggests there's a Goldilocks length for a muscle to generate the most force.
This goes right back to the sliding filaments. It's a direct consequence of that structure. Think about our hand analogy again.
Okay, fingers interlaced.
If you push your hands all the way together so your palms are touching and your fingers are completely overlapped.
My fingers are hitting the back of the other hand. They have nowhere else to go.
Exactly. The muscle's too short. The actin filaments are crashing into the Z discs at the other end. You can't generate any more tension because you've literally bottomed out.
Yeah.
There's no more room to slide.
Okay, so what if I do the opposite? I pull my hands far apart, so only the very tips of my fingers are touching.
Now, there's very little overlap. Most of the myosin heads are just clawing at empty air. They can't find a piece of the actin rope to grab, so the tension you can generate drops to almost zero.
So the maximum strength happens somewhere in the middle.
Yes.
Wow.
There is an optimal resting length where the overlap is perfect. Every single myosin head has a piece of actin it can grab onto. That's where you get the maximum number of cross bridges and therefore maximum tension.
And Ganong points out that our skeletons and the way our muscles are attached are actually designed to keep our muscles operating near this optimal zone most of the time.
The hamstrings are a perfect example. They're biarticular. They cross both the hip and the knee joint. When you run, as you flex your hip, which would shorten the muscle, you're also extending your knee, which lengthens it. The net effect is that the hamstring stays at a relatively constant, optimal length for powerful contractions throughout the whole gait cycle.
That is incredible engineering. We're biomechanically optimized for leverage.
We absolutely are.
Now, what about gearing? If I need to lift something heavy versus something light, how does the body decide how much muscle to actually use?
This is the concept of the motor unit, and crucially, the size principle of recruitment. First, the definition. A motor unit is a single motor nerve from the spinal cord and all the individual muscle fibers it talks to.
It's a squad.
It's a squad. They all contract together. But the size of the squad varies dramatically depending on the job. In a muscle that controls your eye, where you need to track a tiny fly moving through the air, one nerve might control only 3 or 4 muscle fibers.
That's insane precision.
It's microscopic control.
Yeah.
But now think about your quadriceps muscle in your thigh. One nerve there might control 1,000 or even 2,000 fibers. That's a sledgehammer. You don't need fine motor control to stand up from a chair. You just need raw force.
So when I decide to move, does my brain just yell, go, at all the units at once?
No, it whispers first, then it talks, then it shouts. This is the size principle, which Guyton explains beautifully. The spinal cord always recruits the smallest motor units first. These are the ones controlled by the smallest motor neurons, which happen to innervate the type I fibers.
The marathon runners.
Exactly. The small, slow-twitch oxidative fibers. They're very resistant to fatigue. They're perfect for low-level sustained activity, like maintaining posture. You're using them right now just to sit up straight.
But if I decide to stand up and lift a heavy couch?
Then your brain sends a stronger signal down the spinal cord. This louder signal is now able to excite the larger motor neurons, which control the big, powerful motor units. The type 2 fibers.
The sprinter.
The fast-twitch glycolytic fibers. They are huge, powerful, but they burn through their fuel rapidly and fatigue quickly.
Why do it in that specific order? Why not just fire everything at once for a heavy lift?
Smoothness. It allows for a smooth gradation of force. If you fired the big, powerful units first for every single action, every movement would be a violent jerk.
Yeah.
By starting small and layering the bigger units on top as needed, You can go from petting a cat to lifting a barbell with the same muscle smoothly and under control.
That makes perfect sense. It's like a dimmer switch for force rather than a simple on-off toggle.
Precisely. And talking about those fiber types, the classic red versus white muscle is so important for understanding training and performance.
Red muscle is type I, right? Slow twitch.
Yes, it's physically red because it's packed with myoglobin, which is a protein that holds oxygen, and tons of mitochondria, which use that oxygen. It has a rich blood supply. It's built for endurance. Think of the dark meat on a chicken's leg.
And white muscle is type 2, the fast twitch.
Yes, like the chicken breast meat. It's white because it has less myoglobin, fewer mitochondria, and less blood supply. It's built for explosive, brief power, like flapping its wings to escape a predator. It relies on glycolysis breaking down sugar without oxygen.
Which produces lactate.
And acid, which is one of the main reasons why those fibers fatigue so quickly.
So can we change them? If I, a marathon runner, start training for sprints, can I turn my type I fibers into type 2 fibers?
Ganong discusses this concept of plasticity. You can't really change a type I fiber into a type 2 fiber entirely. Your genetics determine your baseline percentage of each. But you can definitely change their character. Your type I fibers can become more powerful, and your type 2 fibers can become more resistant to fatigue.
And you can certainly make them bigger, hypertrophy.
Right. And it's important to understand what hypertrophy is. You aren't growing new muscle cells. For the most part, you are packing more contractile proteins, more actin and myosin filaments into the existing cells. You're adding more lanes to the highway. That's why the muscle gets thicker and stronger.
Okay, so that is the skeletal machine. It's a masterpiece of voluntary speed and graded power, but it has limits. It gets tired. Now we have to pivot completely and look at the machine that cannot fail. The heart.
Cardiac muscle. The reliable pump.
Under the microscope, it looks striated too, right? It has sarcomeres.
It does. On a molecular level, it's using the same actin-myosin sliding filament mechanism we just discussed. But the cellular architecture, the way the cells are put together, is totally different. Because the goal is totally different.
The goal isn't precision. The goal is coordination.
Absolute coordination.
Yeah.
Skeletal muscle is made of discrete, isolated fibers. You can twitch one tiny part of your bicep, but you never, ever want to twitch just part of your heart.
That's fibrillation, a bag of writhing worms, basically. No blood gets pumped.
Exactly. To effectively pump blood, the entire chamber of the heart, the atrium or the ventricle, must squeeze as a single coordinated unit. So the cells are physically and electrically fused together by structures called intercalated discs.
And what's inside these discs that makes that happen?
Gap junctions. These are literally little protein tunnels that connect the cytoplasm of one cell directly to the next. They allow ions, and therefore electrical current, to flow freely from one cell to all of its neighbors.
So if one cell fires an action potential,
they all fire. It spreads through the network like a wave. The heart acts as what we call a functional syncytium.
a together cell.
Yes, for any given chamber, it's an all-or-nothing event.
Now, the electricity itself, the action potential, behaves very differently here. In skeletal muscle, the spark is incredibly quick, a millisecond or two, zap, done. But in the heart, there's this weird, long plateau.
This is one of the most critical concepts in cardiac electrophysiology. When a cardiac muscle cell depolarizes, it doesn't just spike up and come right back down. It spikes, and then it stays high, depolarized, for 200 to 300 milliseconds.
That's an eternity in nerve time. What's holding it up there?
It's a beautiful dance of different ion channels. First, you have the fast sodium channels, same as in a nerve that cause the initial rapid spike up. But then, as Guyton explains, a different set of channels opens, the L-type calcium channels. L stands for long-lasting. They open slowly and stay open.
So positive calcium ions are flowing into the cell.
And at the same exact time, some of the potassium channels that would normally let positive charge out of the cell to bring the voltage back down, they close.
So you're pumping positive charge in and you're trapping the positive charge that's already there.
Exactly. You are actively forcing the cell to stay depolarized, to stay in a contracted state.
Why? What is the evolutionary benefit of holding that contraction for so long?
There are two massive reasons. One is simple mechanics. To pump blood out of a chamber requires a sustained squeeze, not a brief twitch. You need time to eject the fluid. But the second reason is even more important. The absolute refractory period.
Explain that.
As long as the cell is depolarized during that long plateau, its voltage-gated sodium channels are inactivated. It cannot fire another action potential no matter how strongly you stimulate it. It is electrically numb.
So you can't double tap it. You can't send signals too close together.
You cannot. In skeletal muscle, If I zap it with electricity fast enough, the individual twitches merge together into a smooth, sustained contraction. That's called tetanus, a cramp. That's how you hold a heavy box.
But if you tetanize the heart,
It would just freeze in the squeeze position and never relax.
and blood flow would stop.
Instantly,
Yeah.
you would die. The plateau ensures that the action potential lasts almost as long as the contraction itself. It forces the heart to fully relax and have time to refill with blood before it's even possible for it to get another signal to beat. It is a chemical and electrical fail-safe against cramping.
That is just genius. The very chemistry of the cell dictates the fundamental rhythm of the pump.
It does.
Now, remember that bathtub plug mechanism in skeletal muscle? The direct mechanical yank? Ganong is clear that the heart doesn't do that.
No. The heart uses a more elegant system, chemical triggering. It's called calcium-induced calcium release, or CICR.
Okay, how does that work?
In the heart, the dihydropyridine receptor, that voltage sensor on the T-tubule, is not physically touching the ryanodine receptor on the SR. Instead, when the action potential comes down, the DHPR opens and acts as a calcium channel itself. It lets a small splash of calcium into the cell from the outside.
From the fluid inside the T-tubule.
Right. This small amount of trigger calcium then diffuses across the tiny gap, binds to the ryanodine receptor on the SR, and that binding is what tells the main floodgate to open.
So a little bit of outside calcium pulls the trigger for the massive release of inside calcium.
Exactly, and this has a massive clinical implication. Skeletal muscle brings its own starter fluid, essentially. It relies almost entirely on its internal SR calcium stores. It doesn't really care what the calcium level in your blood is.
Not the heart.
The heart needs that trigger calcium from the outside to beat strongly. The strength of its contraction is directly related to the extracellular calcium concentration. If your blood calcium drops too low, a condition called hypocalcemia, your heart gets weak and floppy.
That's a critical distinction. The heart is much more dependent on the wider body environment.
It is, very much so.
One last thing on the heart, the Frank-Starling mechanism. We talked about the length-tension relationship in skeletal muscle. How does the heart use that same principle for self-regulation?
This is the heart's intrinsic cruise control. The law is simple. The heart pumps what it receives. It automatically adjusts its output to match its input.
So if I go for a run, my leg muscles pump more blood back to my heart. More blood flows into the ventricle.
So the heart fills up more before it beats. The muscular walls of the ventricle stretch. Now think back to the sarcomeres inside those muscle cells. By stretching the wall, you are pulling those actin and myosin filaments into a better, more optimal overlap.
You're moving them from a shorter length into that Goldilocks zone of the length-tension curve.
Exactly. So simply by virtue of being stretched, the heart mechanically arranges its filaments to grab on better, form more cross bridges, and therefore contract with more force on the very next beat.
So it doesn't need a signal from the brain to tell us, hey, more blood is coming, pump harder.
No. The physics of the fibers handles it automatically. It's like a rubber band. The more you stretch it within limits, the harder it snaps back. It's a beautiful, elegant, self-regulating system.
Beautiful. Okay. Skeletal, the drag racer. Cardiac, the reliable pump. Now we turn to the rebel of the family. The weird one. Smooth muscle.
The adaptable squeezer.
A critique we got from some listeners on a previous deep dive was that we kind of rushed this topic, so let's give smooth muscle its proper due. Because it is weird.
It breaks all the rules we just established. First, and most obvious, no striations.
No stripes.
Which means the sarcomeres aren't lined up in those neat crystalline rows. In smooth muscle, the actin and myosin filaments are arranged in a crisscrossing web, like a net. They're anchored not to Z-lines, but to structures called dense bodies.
What's a dense body?
You can think of it like a knot in a fishing net, either floating in the cell or attached to the membrane. The filaments pull between these knots, so when the cell contracts, it doesn't just shorten neatly, like a telescope, it scrunches up in all directions, kind of like a raisin.
And because of that less rigid structure, it can shorten a lot more, can't it?
A huge difference. Skeletal muscle is limited to shortening maybe 30% of its resting length. Smooth muscle can shorten up to 80%. Think about your bladder. It has to go from a tiny, empty walnut to a large, full grapefruit. A rigid, striated skeletal engine couldn't handle that kind of geometric change.
Okay, now for the switch. The regulation. No troponin.
No troponin. This is the fundamental shift in logic. Smooth muscle doesn't bother with covering the binding sites on the actin. The actin is always ready, always available.
So why isn't it always contracting then? What's the safety catch?
Because the myosin is asleep. In skeletal muscle, we move the guard troponin out of the way of the rope. In smooth muscle, we have to wake up the worker, the myosin.
How do you do that?
It's a chemical cascade. Calcium enters the cell, mostly from the outside. The SR is pretty rudimentary in smooth muscle. That calcium binds to a protein called calmodulin.
Calmodulin.
Calcium calmodulin team then goes and activates an enzyme called myosin light chain kinase, or MLCK.
Okay, a kinase is an enzyme that adds a phosphate group to something, right? It's like adding an energy packet.
Exactly. MLCK phosphorylates a part of the myosin head called the light chain. This is the alarm clock. This phosphorylation event is what allows the myosin head to finally interact with actin and start the cross-bridge cycle.
This sounds a lot slower. You have diffusion, binding, enzyme activation, phosphorylation.
It is much, much slower. The whole process can take seconds, not milliseconds, but the trade-off you get for that slowness is incredible efficiency. And this is the part I want to drill into, the latch mechanism. Guyton highlights this as the secret to its energy saving.
This is how your arteries can maintain blood pressure all day long without you passing out from metabolic exhaustion.
That's exactly what it's for.
So how does it work?
Okay, so normally, to let go of actin, the myosin head needs a fresh ATP.
Right, ATP to detach.
In smooth muscle, once the myosin is attached to actin and is holding tension, another enzyme, a phosphatase, can come along and snip that phosphate group off the myosin head while it is still attached to the actin.
So it deactivates the myosin motor while it's still holding on.
Precisely. And when you do that, the myosin head's affinity for actin skyrockets, but its cycling rate plummets. It essentially gets stuck. It becomes like a rusty hinge. It stays clamped to the actin, maintaining tension, but it stops cycling through ATP.
It's a molecular parking brake?
It is a perfect analogy. It's a parking brake. It can hold that squeeze for hours, maintaining tone, using as little as 1/1300th the energy that skeletal muscle would need to do the same job.
That is just brilliant. It allows the body to maintain tone in places like blood vessels or sphincters, essentially for free.
Correct. It's all about sustained, low-cost contraction.
And there's one final bizarre feature, stress relaxation, the bladder trick.
Right. If you take a rubber balloon and start filling it with water, the pressure inside rises steadily and it stays high. But if you fill a bladder, the pressure goes up for a moment and then it drops back down, even as you keep filling it.
Why? What's happening?
Because the smooth muscle filaments can physically detach, slide past each other, and re-anchor themselves in a new, longer configuration. They literally reset their own resting length to accommodate the new volume.
So the bladder wall gets bigger without necessarily feeling tighter.
Up to a point, yes.
Yeah.
It's what allows you to store a significant amount of urine without a constant, screaming urge to pee until you finally hit that critical stretch limit.
It really is the most adaptable and plastic of all the muscle tissue.
By far, and its control systems reflect that. Skeletal muscle has one boss, the voluntary motor nerve. Smooth muscle listens to everybody.
Nerves.
The autonomic nervous system, yes, but also hormones, epinephrine, oxytocin, angiotensin. Also local chemistry. A lack of oxygen, too much CO2, or high acidity can all cause smooth muscle and blood vessels to relax, which is vital for regulating local blood flow.
And just stretch itself.
And stretch. It's an incredibly versatile tissue.
So we've crossed the landscape. We've seen the rigid voluntary power of skeletal, the rhythmic fail-safe reliability of cardiac, and the efficient, adaptable plasticity of smooth.
3 different engines built from one core set of parts.
If we have to synthesize all of this into a so what for the listener, what is the big unifying takeaway?
I think the first theme has to be calcium is king. Nothing moves without it. It is the universal trigger. But the way each tissue handles calcium, whether it's a direct mechanical pull, a chemical trigger, or an enzyme activation cascade, that defines the tissue's destiny and its role in the body.
And the second theme?
Form follows function. It's the classic biological principle. You can see the mission of the muscle just by looking at its structure. The gap junctions in the heart scream unity. The latch mechanism in the gut screams endurance. The T-tubular system in the leg screams speed.
It really makes you look at your own body differently. We aren't just a bag of meat. We are precision-engineered machines adapted for specific tasks.
And here's a final provocative thought to leave you with to build on that.
Go for it.
We tend to treat these systems as fixed machines, like a car engine. You buy it, and that's the engine you have. But muscle isn't like that. It is profoundly plastic.
What do you mean?
If you start sprinting every day, your skeletal muscle literally rebuilds its proteins to be faster and more powerful. If you develop high blood pressure, your heart muscle thickens, hypertrophies. to fight against that increased resistance. If a woman becomes pregnant, the smooth muscle of her uterus grows from a tiny organ to one capable of the most powerful contractions in the human body.
The machine rebuilds itself while it's running.
Constantly.
Yeah.
It remodels itself based on the stress you apply to it. Our bodies are not just machines. They are machines that are perpetually under construction, constantly adapting to the demands we place on them.
I love that. A machine that rebuilds itself. Listeners, if you want to really visualize this stuff, especially the interlaced fingers of the sarcomere or the length tension curve, please go open the books. Read the excitable tissue muscle chapter in Ganong. Look at the beautiful graphs and diagrams in Guyton.
The visuals really make the physics click in a way that just listening sometimes can't. This is a very visual topic.
It really is. Thanks for diving deep with us on this. We'll see you next time.
Keep learning.